Hi everyone, this is Dr. Ross. This video will be exploring the origins of vertebrate immune cells. It is important to understand the mechanisms that guide this process before diving into the more intricate workings of the immune system. So remarkably, all mature blood cells arise from a single type of cell called a hematopoietic stem cell through a process called hematopoiesis. The process of hematopoiesis begins before birth, actually, and takes place primarily in the yolk sac and liver before eventually transitioning to the red bone marrow. This happens at around 20 weeks of gestation. Although red bone marrow is found in many bones, hematopoiesis is most active in the vertebra and pelvis, the sternum, and ribs. The reason hematopoiesis occurs in the bone marrow is because this is where the progenitor cells that give rise to all mature blood cells resides. So the progenitor of all blood cells is the hematopoietic stem cell, or HSC. HSCs are not common, and their numbers are very strictly controlled and tightly regulated by signals that they receive in the bone marrow. Like all adult stem cells, HSCs spend most of their time in a type of suspended animation called quiescence. And this is just an altered metabolism that helps the cells survive for extended periods of time in the hypoxic environment of the bone marrow. These types of cells are referred to as long-term HSCs. Now, when we have cell damage or cell death, the hematopoietic stem cells will be provoked to exit quiescence and begin actively dividing again. Now, when the appropriate signals are present, the long-term HSCs will give rise to what are called short-term HSCs. And these will develop and proliferate into multipotent progenitors, or MPPs. Now, MPPs have a very limited ability for self-renewal, but they proliferate rapidly. Now, some of these cells will become common myeloid progenitor cells, which can further differentiate into innate immune cells and red blood cells. Other MPPs will become common lymphoid progenitor cells, and these will give rise to cells that play a role in the adaptive immune system. Now, these populations of cells can be identified by the presence of certain surface markers as well as the absence of others. So, for example, HSCs can be identified from other cells by the presence of the surface markers SCA1 and c-KIT. MPPs also contain these receptors as well as another receptor called CD34. Now, because HSCs and MPPs have not committed to a specific lineage, they are also termed Lin-minus. Now, MPPs also express, MPPs that will express the surface receptor FLT3 will actually become destined to become the CLPs, or common lymphoid progenitors. CLPs also express a surface receptor called ILR7. Now, hematopoiesis is controlled by many different transcription factors, and these factors will drive the process of differentiation into the different blood cell types. So more regulators are involved than I'm going to talk about, but some of the key ones are GATA1, and this is going to promote erythrocyte and megakaryocyte development. PU1 guides myeloid lineage differentiation. Icaros is critical for lymphoid lineage specification. And then it's not just the presence or absence of these transcription factors that is important, but also the balance. So for example, the balance between transcription factors like GATA1 and PU1 will also help determine lineage commitment. In addition to transcription factors, the the blood cell production, maturation, and function is controlled by the presence of certain growth factors and cytokines as well. So conceptually, these types of molecules can be divided into two groups. You have your multilineage, or early acting growth factors, which stimulate multiple lineages. So SCF, which is stem cell factor, this is going to play a crucial role in the survival, proliferation, and differentiation of the HSCs and progenitor cells. And it's known to synergize with FLT3 and IL-7. IL-7 is essential for the development and proliferation of lymphoid progenitors, particularly the early B and T cells, and it can be found in the thymus. FLT3 ligand or FLT3L also plays a significant role in supporting differentiation of early B cells as well as ILCs or innate lymphoid cells. We also have GM-CSF. This is going to act on the HSCs as well, and it's going to usually promote proliferation into granulocytes and macrophages, and it can also influence other myeloid substrates, subsets. IL-3 acts on early hematopoietic precursor cells. Here. And it also, IL-6 also enhances proliferation of hematopoietic stem cells as well. And it is stimulated by IL-3. The other group of lineage-specific growth factors are called, the other group are called lineage-specific growth factors, and these stimulate differentiation and survival of a single lineage. So lineage-specific growth factors are signaling molecules. They're often cytokines, and they guide the developing cell toward a very particular type of mature cell, like a red blood cell. Granulocyte colony stimulating factor, which is up here, promotes the production of granulocytes ultimately. Macrophage colony stimulating factor. Promotes macrophage maturation, and then TPO or thrombopoietin produces platelets, and EPO or erythropoietin will influence the production of erythrocytes. Hematopoiesis is a very complicated process in summary, and it receives many internal and external clues or cues that give rise to a variety of cell types. Hematopoiesis, so in summary, hematopoiesis is very multifaceted. It's a complicated process involving many internal and external cellular processes, and this ultimately gives rise to a variety of cell types that are players of the immune system. So during this process, different surface receptors appear on different cells during development, allowing them to bind and respond to growth factors in the environment. Ultimately, the response to these different growth factors will contribute to how the cells develop. Finally, transcription factors also play an important role as well, because different transcription factors will drive different patterns of gene expression. And that is it for our hematopoiesis part 1.